Welcome to BioBits and Bytes. We are diving deep today into one of the most compelling and potentially civilization-changing fields at the intersection of chemistry, biology, and engineering. We're talking about the quest to turn raw sunshine directly into dense, storable chemical fuel using microscopic factories.
It's a fundamental challenge,
Hmm.
and it's really driven by global demand. I mean, if you look at the projections, global energy need is expected to increase at least twofold by the middle of this century.
Yeah.
We all know the energy solution is solar, but the scale of the challenge is just massive. Not in terms of raw supply, but in, managing the flow.
Yeah, we often hear that truly staggering fact that just one single hour of sunlight hitting our planet holds enough energy to cover absolutely everything humankind uses in an entire year.
It's an unbelievable reservoir.
An unbelievable reservoir of power, but it's wildly inconsistent, right?
Exactly.
Yeah.
The challenge isn't capturing the light. I mean, that's what highly efficient photovoltaic or PV cells do really well.
Hmm.
The challenge is energy storage. How do you take that colossal, instantaneous input of solar power, which, only happens during the day, and store it efficiently?
And in a form that's high density, easily transportable, and available on demand day or night for planes, trucks, or rockets.
Yes.
It's the famous analogy, right? Trying to pour the entire ocean into a teacup, you just need a better vessel for storing that energy.
You do. A lithium battery is often too heavy for high-density applications like flight, and converting solar into heat is often inefficient.
So what's the better vessel?
And that better vessel is what we are calling artificial photosynthesis, or APS, and its engineered biological application, microbial synthetic photosynthesis, or MSP.
Okay.
This deep dive is about literally scripting sunlight. We're talking about building solar batteries, not out of heavy metals, but out of chemical bonds, mimicking, and this is the critical part, surpassing nature's own efficiency limits.
So our mission today is to unpack the sources that highlight these revolutionary breakthroughs, and we're going far beyond simple plant-based bioethanol here.
Oh, way beyond.
We're looking at systems demonstrating lab-based solar conversion efficiency gains that hit up to 17.8%. That is a massive
A
leap.
huge leap.
And we're going to detail how synthetic biology is building microbes capable of producing high-performance liquid rocket fuel and biodiesel.
We're moving into a realm where the chemistry is engineered for specific performance metrics. We're focusing on energy density that rivals or, even exceeds the best fossil fuels.
Okay, let's unpack this. And I think we should start with why nature, the original solar factory, isn't already doing this job for us more efficiently.
That's the perfect place to start. Natural photosynthesis, performed by everything from towering redwoods to tiny cyanobacteria, is beautiful, it's robust, and it's absolutely crucial for our biosphere. But it is fundamentally inefficient when you look at it purely as a power plant.
Yeah.
It's optimized for survival, for growth, and for reproduction, not for maximal fuel production.
So what does that inherent inefficiency look like in hard numbers, and what causes it?
Well, the constraints are systemic, and they're deeply rooted in the biochemistry. Plants, on average, achieve only about 1% of the theoretical thermodynamic maximum efficiency when you measure it annually across the globe.
1%.
Just 1%. Even during a perfect peak growing season, the efficiency generally ranges from a pretty narrow 1.4% to maybe 2.0%.
That is an astonishingly low number, especially when you consider the sheer potential of solar energy that's available.
Yeah.
So what exactly is going wrong inside the plant cell that limits it to 2%?
There are a few key bottlenecks. We can explain the core mechanism using what biochemists call the Z scheme, but the biggest biological limitations are really photorespiration and the sheer complexity of the whole process.
Let's simplify the Z scheme first, maybe with that assembly line analogy.
Perfect. Imagine photosynthesis as this massive, multi-stage, light-powered conveyor belt.
Okay, so the conveyor belt starts at a place called photosystem 2, where the first burst of solar energy of photon strikes, and this is the heavy lifting part, right, where water is split.
That's right. It splits water, H2O, to release electrons, and crucially, oxygen as a byproduct. And that is a tough 4-electron process that requires a big energy boost from that first photon.
Right, those electrons start traveling down the chain, which is like a small water slide, releasing a little energy as they go.
Exactly.
Yeah.
And that energy is used to make the cell's universal currency, a molecule called ATP. But by the time they finish that slide, the electrons are just too low in energy to do the final job, which is fixing carbon.
So they need a second major energy boost to finish building the fuel.
Which is where photosystem I comes in. It absorbs a second photon to lift those electrons to a much, much higher energy state.
And that's essential.
It is. This second boost is essential to generate the ultimate reducing power, a high-energy molecule called NADPH. And it takes both of those NADPH and ATP, both products of those two light hits, to enter the Calvin cycle and actually convert CO2 into sugar.
So, if I'm understanding correctly, nature's system is a bit like a clunky assembly line that requires two separate light boosts, 2 photons per electron pair, just to make the necessary chemical energy needed to convert CO2 into sugar. Is that the fundamental inefficiency?
That's the kinetic inefficiency, yes. But there's a biological one too, and it's a big one, photorespiration.
Okay.
The main enzyme responsible for carbon fixation, it's called rubisco, is notoriously poor at differentiating between CO2 and oxygen.
So it makes mistakes.
Constantly. When it accidentally binds oxygen, it starts this incredibly wasteful process called photorespiration that literally undoes the work of photosynthesis. It releases CO2 back into the atmosphere and consumes energy. It's a huge problem, especially at high temperatures.
So between the need for 2 photons and this constant chemical confusion, the natural world is just stuck below 2% efficiency.
Precisely. And if we rely solely on natural systems, like growing vast fields of corn or algae for all our energy needs, that inefficiency means we need enormous amounts of land.
Which then competes fiercely with food production, fresh water, and, ecological preservation.
It does. It raises a huge challenge.
I see it. But demanding a tenfold increase in efficiency, say 10% versus 1%, that sounds less like engineering and more like a complete scientific redesign. What kind of target do we really need to hit to make this competitive?
To significantly reduce that land area requirement and become a true large-scale energy source, our sources define a critical threshold. And artificial technology really needs to achieve 10% efficiency or better.
10%.
Below that, the land use requirements just become prohibitive. So 10% is the goal for massive, sustainable impact.
That gives us a clear goal, at least 10 times better than a plant on an industrial scale. So how are researchers trying to achieve this by moving into the pure chemistry of artificial photosynthesis, or APS?
APS is the strategy of constructing an artificial leaf, if you will, entirely out of synthetic components, catalysts, light absorbers to capture solar energy, convert it, and store it efficiently in high-energy fuels like pure hydrogen gas.
So this often involves biomimicry, stealing the best ideas from nature, but making them faster, tougher, and less prone to those mistakes like photorespiration.
Yes, and sometimes we use those biological parts directly as catalysts, which is fascinating. Organic chemists are already pioneering techniques using what's called visible light photocatalysis, which is directly inspired by photosynthesis.
Can you give us a tangible example of that, where they use a biological part to drive a synthetic reaction?
Certainly. One very practical and sustainable example involves using visible light and oxygen in combination with the natural pigment chlorophyll.
The actual molecule that captures light in plants.
The very same one. They use it as the photosensitizer. This low-cost, mild, natural setup can be used to produce complex organic molecules called tetrahydroquinolines.
And why is using chlorophyll such a big deal, beyond just being green?
Because it lets chemists perform these highly complicated multi-step synthetic reactions from simpler starting materials under incredibly mild conditions, just visible light and atmospheric oxygen. You avoid using expensive, toxic, or rare metal catalysts. It really shows that even the simple light-capturing ability of natural pigment can be integrated into high-efficiency, sustainable processes.
So we've got the vision, surpass 10% efficiency, and we know we can use the chemical rules of light capture, even using natural pigments to drive reactions. But the real revolution seems to be when we stop copying nature entirely and start rewriting the biological code itself. I think that brings us to microbial synthetic photosynthesis.
This is where we transition into the definition of microbial synthetic photosynthesis, or MSP. It's really the next frontier in synthetic biology. It uses advanced metabolic engineering and synthetic biology tools to equip microorganisms with entirely optimized non-native pathways.
Okay, let's dig into the mechanics of that. So we're basically taking a microbe's existing metabolic machinery, its internal assembly line, and we're rewriting the instructions to make specific high-value fuels instead of just, making more of itself.
Precisely. We are reprogramming them to efficiently take sunlight, water, and CO2 and turn them directly into targeted energy products, whether that's hydrogen, advanced hydrocarbons, or, as we'll see, super dense liquid fuels.
And they're making jet fuel and rocket fuel, if I'm reading the sources correctly, not just average stuff either.
They are, and this is where the chemistry gets really cool and critically very energy dense. Researchers were specifically hunting for fuel molecules that contain what are called cyclopropane rings.
Okay.
These are 3 carbon rings that are strained when they're bonded, like a coiled spring, and that strain energy translates into a huge punch of potential energy when it's burned.
So they wanted fuel that wasn't just clean, but was actually better than what the aerospace industry uses today.
That was the goal. And they found the initial blueprint in these obscure, hard-to-grow soil bacteria belonging to the genus Streptomyces.
And what did they find there?
They focused on the genes that allow these bacteria to naturally produce a molecule called gosomycin. And gosomycin is noteworthy because it naturally contains 5 of these cyclopropane rings. It was described by the researchers as looking like a jaw filled with pointy teeth.
Gosomycin. From jaw filled with pointy teeth. That must have been a pretty incredible moment of discovery, finding a natural blueprint for what turns out to be a key component for rocket fuel.
It was. The scientists essentially became metabolic chefs. They studied the genes that build gosomycin, and then they remixed that existing bacterial machinery.
So like a baker rewriting a recipe.
Exactly. They kept the core instructions for creating the cyclopropane rings, but they skipped the unnecessary final steps of the original molecule structure.
And what was the result of this precise metabolic remixing?
They created a completely new class of fatty acids that they called fuelamycins, or popamies, that contained up to 7 cyclopropane rings chained onto a carbon backbone. These molecules are the revolutionary fuel candidates.
Wow. And that's where we get those incredible performance metrics. What did the computer simulations suggest for these fuelamycins?
The computer simulations, which were run by collaborators at Sandia National Laboratories, suggested that these processed POP fuel candidates would have energy density values of more than 50 megajoules per liter.
50 megajoules per liter. Let's put that into context because that sounds like a total game changer for flight and space travel.
It absolutely is. I mean, for you listening, conventional gasoline typically sits around 32 megajoules per liter. Jetta, the most common jet fuel, and RP-1, a popular rocket fuel, both hover around 35.
So hitting over 50 is a massive, almost 40% jump in energy density compared to current rocket fuels.
A huge jump. And for rocketry, that density is everything, isn't it? A higher energy density means you need less volume of fuel for the same thrust.
Right, which dramatically improves the mass fraction, the ratio of payload to total launch mass. This would directly translate to increased payload capacity or extended range.
That's the real-world application. It's not just about cleaner fuel. It's about higher performance fuel that changes the entire engineering calculus for aircraft and rockets. But there is a small chemical caveat in the current design.
You mentioned the dead weight oxygen atom.
A small one, chemically, yes.
Yeah.
The current fuelamycins contain 2 oxygen atoms on each molecule. Now, these help in the biological synthesis, but oxygen adds dead weight and doesn't really contribute to the combustion energy.
So the next goal is to get rid of them.
Right. The next major engineering goal is to create a reliable process to remove those 2 oxygen atoms, which should further increase the performance and stability when it's blended into jet fuel.
So that's the advanced fuel side. Now let's pivot to the preferred microbe chassis itself. You mentioned the CO2 fixing workhorses, the cyanobacteria.
Yes, the cyanobacteria, they're often called blue-green algae, though they're bacteria, are the primary chassis for direct solar-to-fuel conversion. They're essentially the green E. coli.
And why are they so attractive?
They use light and CO2 for growth. They have a simple structure, they grow quickly, and we have established sophisticated genetic tools for engineering them. Model organisms like Seneca cystis and Senechococcus elongatus are heavily used in labs.
And since these are photoautotrophs, the tools we use to program them have to be, I imagine, sensitive to light and darkness and allow for extreme precision.
Absolutely. One critical area is promoter engineering. You can think of a promoter as the DNA sequence that acts like an on-off switch, or even better, a dimmer for a gene.
And to maximize fuel production, you need really strong promoters to drive high expression of those new fuel-making genes.
Exactly. They start with strong native promoters, like one called PPSBA2, which is naturally light-regulated. But synthetic biology lets us improve on nature's reliability and strength.
So how much control can they actually gain?
By systematically modifying native promoters, scientists can create synthetic variants with highly improved and predictable activity. For example, modifying one native promoter led to the variant PCPC560, which is super strong. Even more remarkably, researchers created a library of promoters regulated by an inducer molecule, and the best one showed a massive 290-fold change in gene expression.
A 290-fold change. That is significant leverage at the molecular level. Does that level of precise control essentially eliminate the need for those naturally strong but less predictable native promoters.
That's a great question. In a highly controlled bioreactor, yes. That precise, inducible control allows engineers to allocate the cell's resources. First, you grow the cells, you build up the biomass. Then, once the culture is dense, you flip the switch and dedicate all available energy toward making fuel.
And what other high-precision tools are they using inside these green cell factories?
Another fascinating tool is the use of riboregulators. These are clever RNA-based systems made of two parts, a cis-repressed RNA and a trans-activating RNA. They act as simple, reliable genetic switches right at the level of translation.
So how does that work in practice? Is it like a literal molecular lock and key mechanism?
That's a perfect analogy. The lock is designed to bind to the ribosome binding site on the target gene's message. By binding there, it physically blocks the ribosome, the cell's protein-making machine, from starting translation. So the fuel-making gene is OF.
And the key.
The key is the trans-activating RNA. When it's introduced, it binds tightly to the lock, which relieves the blockage, freeing the ribosome binding site and flipping the translation switch on-in. It's an elegant, energy-efficient, RNA-based control system.
That's incredibly precise. And since the core function of cyanobacteria is fixing CO2, I understand scientists are also targeting the internal machinery for carbon fixation itself, specifically the carboxysomes.
The carboxysomes are truly marvels of natural nanotechnology. They are these protein nanocompartments found inside cyanobacteria that serve one crucial purpose. They act as microscopic pressure chambers to concentrate CO2 around the enzyme rubisco.
Which, as you said earlier, is notoriously sluggish and confuses CO2 with oxygen. So the carboxysome is evolution's ingenious workaround to solve that photorespiration problem.
Exactly. By using the carboxysome shell to actively pump and trap CO2 inside, the local concentration of carbon dioxide can be hundreds or even thousands of times higher than the surrounding cell. This dramatically boosts rubisco's efficiency.
And what's the high-stakes goal of engineering these carboxysomes?
The big prize is successfully transferring them into agricultural crops like rice, wheat, or corn. The goal is to dramatically increase crop carbon fixation efficiency globally. Imagine if our food staples could perform photosynthesis at 3 or 4 times their current rate.
The potential application for global food production is enormous, but I imagine getting an entire nanocompartment to integrate and function inside a plant is a massive undertaking.
It is. The work is very challenging.
So we've gone deep into how they control the cell's internal structure and chemistry. But these green cell factories also have to contend with an external biological rhythm, the sun rising and setting.
This is the major metabolic complexity challenge for any outdoor photobioreactor. All cyanobacteria have a circadian clock anchored by master regulator proteins, Kaya, Ki-B, and Kaya. These proteins force the organism to alternate between day and night metabolisms.
Can you give us an example of how that biological clock complicates fuel production?
Well, consider Cyanothus 51142. It dedicates its day cycle to fixing carbon and producing oxygen through photosynthesis. But at night, when respiration reduces intracellular oxygen, it switches its focus entirely to fixing nitrogen and sometimes uses a hydrogenase to produce hydrogen fuel.
So when we are trying to model or predict fuel production using a tool like flux balance analysis or FBA, this constant change makes it hard to assume a pseudo-steady state.
Exactly. FBA, a crucial tool for metabolic engineering, assumes that the system is relatively stable over time, but the need to incorporate light as a diurnal variable and differentiate between light and dark metabolisms poses a significant challenge for its predictive power.
So the key engineering takeaway isn't just that the clock exists, it's that if you try to run a high-volume fuel factory outside, you can't force the cell to fix carbon when its natural rhythm is trying to switch to something else.
You have to design the synthetic circuit to be robust against, or even smarter, integrate with that circadian rhythm to maximize output during the most efficient cycle windows. It's a critical design constraint.
This inherent constraint, the low efficiency of natural light capture and the complexity of these internal biological cycles, seems to be what drove scientists to the most exciting breakthrough we found: the hybrid system that completely removes the microbe from the job of capturing light.
Yes, the bioinorganic hybrid system is a strategic move to decouple the inefficient parts of natural photosynthesis, like light capture, from the very efficient parts of synthetic biology, like precise CO2 fixation.
Which leads to what's often called electromicrobial production, or EMP.
Exactly.
Can you define EMP for us simply?
EMP is a two-step system. Step 1. We use an external energy source, typically electricity from highly efficient solar PV panels, to create chemical substrates, usually pure hydrogen gas, H2.
Mm-hmm.
Step 2, we then feed that high-energy H2 to engineered bacteria called chemoautotrophs, which are incredibly efficient at fixing CO2 using that supplied chemical energy.
So the efficient non-living KV panel captures the light at, say, 35% efficiency, and the microbe just does the part it's good at, which is fixing carbon, using that guaranteed energy. It's an end run around natural photosynthesis' low efficiency.
It is, and the key host here is the H2 oxidizing bacterium, or HOB, specifically Ralstonia eutropha H16. It's sometimes called a null gas bacterium.
And it naturally uses hydrogen and oxygen to grow.
Right. It naturally uses H2 and O2 to fix CO2 for growth, making it a perfect gas-guzzling bug. By engineering this strain, the chemical energy in the solar-derived H2 can be directed toward making liquid fuels like butanol or isopropanol instead of just making more bacteria.
Tell us about the landmark integrated microbial inorganic system, the artificial leaf achievement that really put up those record-breaking numbers.
This system ingeniously combined robust, earth-abundant inorganic catalysts with the microbial fuel factory. The setup essentially acted as a water-splitting reactor with a water-splitting anode and cathode.
What were the catalysts used for the water splitting? Because often traditional electrolysis uses expensive, rare metals like platinum.
That's a critical difference, and it touches on the economic viability. The anode, where oxygen is produced, used cobalt phosphate, or COPI. And the reason this COPI is a big deal isn't just efficiency, it's cost. It's earth-abundant.
Unlike platinum or iridium.
Right. It's also a low overpotential catalyst for the oxygen evolution reaction, meaning it splits water efficiently at a lower voltage. The cathode used a nickel-molybdenum-zinc alloy, or stainless steel mesh, to generate the H2.
So the use of earth-abundant materials significantly lowers the capital expenditure for massive industrial adoption right from the start. How did the microbe integrate with all this?
The Ralstonia eutropha was cultured directly in the electrolyte solution. The electrodes split the water, producing O2 and H2, and the R eutropha has special oxygen-tolerant hydrogenases, which are key. It oxidized the H2 and used that energy to generate the necessary N, ADPH, and ATP to power the Calvin cycle, fixing the CO2 into products.
Okay, we have to talk about the efficiency data now because it is truly revolutionary in this context.
It is the headline number that changes the field. The wild-type R eutropha H16, driven by this bioinorganic system, achieved a maximal bioelectrochemical efficiency of 17.8% for biomass production.
Okay, let's pause and simplify that part again. Natural plants are peaking around 2% solar efficiency, but this bioinorganic system, by decoupling the light capture from the CO2 fixation and feeding it pure hydrogen energy, jumped up to almost 18% efficiency for just creating basic cellular biomass.
Precisely. That 17.8% demonstrated that these hybrid systems can offer yields far beyond what photosynthetic organisms can achieve on their own. The high efficiency is thanks to the efficient catalysis, but also because this system avoids generating reactive oxygen species, or ROS, which stress and kill the cells in direct photobiological systems.
That's biomass. What happened when they engineered the microbe to make a desirable liquid fuel?
They engineered a strain that produced isopropanol fuel, a simple alcohol, and that achieved a maximal bioelectrochemical efficiency of 3.9%. Now, that's lower than the biomass number, but this yield of 216 milligrams per liter was the highest reported solar to isopropanol yield at the time.
What's fascinating here is the trade-off. We have solar to electricity rates from commercial PV panels that might be, 39.5% or higher, but that electricity requires external, often expensive battery storage. Then we have this 3.9% solar to chemical efficiency for liquid isopropanol. Why accept the lower efficiency?
This is the core economic insight and the value proposition. The 3.9% figure represents energy stored directly in stable chemical bonds. That is, energy that is easily pumped, transported via existing pipelines, and compatible with existing fuel infrastructure.
And it offers long-term, high-density storage capacity without needing massive battery farms.
Exactly. The value proposition has to account for this inherent storage capability, which justifies a lower nominal conversion efficiency than pure electricity generation.
So stability and ease of infrastructure integration is the justification for the lower number.
Exactly. When you factor in the energy lost in battery charging, discharging, and the materials cost of large-scale battery systems, the chemical storage of liquid fuels at 3.9% starts looking incredibly competitive.
Now, briefly, let's consider the alternative direct bio approach we mentioned, generating pure hydrogen gas using cyanobacteria. H2 is the simplest solar fuel, but why is it so hard to harvest directly from photosynthetic organisms?
The fundamental constraint, and it's a persistent challenge, is O2 sensitivity. The hydrogenase enzymes that produce H2 are extremely sensitive to oxygen. They're often poisoned by it.
Hmm.
Since photosynthetic organisms like Anabaena are performing oxygenic photosynthesis, meaning they're splitting water and making O2 as a byproduct, the cell is constantly producing the inhibitor right next to the enzyme that needs a low oxygen environment.
It's an internal paradox. The system that uses light to generate the electrons is also poisoning the enzyme that produces the final fuel. What are nature's workarounds?
Nature has two main strategies. Some organisms, like Anabaena, solve this by creating special cells called heterocysts. These are non-photosynthetic cells that protect the enzyme from oxygen.
And the other way.
Other species, like Cyanothase 51142, temporarily separate these processes. They fix carbon during the day, and then they use the dark cycle, when respiration naturally reduces intracellular oxygen levels, to engage in hydrogenase activity.
That temporal separation is a great natural strategy, but it forces an intermittent production cycle. What is the engineering solution to this hydrogen hurdle?
Engineers are focused on two major synthetic biology approaches. First, modifying the structure of the hydrogenase enzyme itself to make it tolerant to oxygen. Or second, engineering a self-regulating circuit that completely shuts down O2 production in the local environment of the hydrogenase.
Despite the constraints, we do see some incredible potential in short bursts. Our sources mention Anabaena variabilis showing very high short-term hydrogen evolution rates, like 68 micromoles per milligram of chlorophyll per hour.
Those high short-term rates demonstrate the intrinsic catalytic power of the enzyme. But the instability, that O2 poisoning, is what causes that rate to crash over time. To scale it up economically, engineers either need to eliminate O2 entirely or succeed in engineering that oxygen-tolerant hydrogenase. This stability issue is one of the key techno-economic bottlenecks.
Let's shift gears from the fantastic science to the cold, hard economics and market reality. The data suggests this isn't just a lab curiosity anymore. The artificial photosynthesis market is actually commercializing.
Absolutely. The market snapshot is impressive. It was valued at around $83 million in 2024 and is expected to hit nearly $285 million by 2033.
And that's a compound annual growth rate, a CAGR, of over 14%.
Right, a 14.62% CAGR. This double-digit growth signals very serious commercial interest, driven by the need for high-density, storable energy solutions.
Before we get to the liquid fuels, let's talk about a major current application of this bioelectricity technology, microbial fuel cells, or MFCs, where these microorganisms are used to generate electricity directly.
MFCs are incredibly fascinating. They utilize microorganisms to convert chemical energy from biomass, often organic waste or wastewater, directly into electrical power. This offers a major dual benefit: low-emission power generation and effluent treatment.
What are the primary real-world uses for MFCs right now?
The applications are diverse. They're primarily used for wastewater treatment, municipal, industrial, and agricultural, while simultaneously generating bioelectricity that can offset some of the treatment plant's energy consumption.
And they're also being explored for biomedical devices.
Yes, for providing low-power, long-term, consistent power to devices placed inside humans, fueled by the body's own biological processes.
That's a wild idea, having a tiny, stable power plant fueled by biological waste inside your body. Did they report any specific performance for the efficiency of these cells?
Yes. Studies on arphaeroducins, a common host, showed it was able to generate electricity with an impressive 80% electron yield. This is a measure of how efficiently the chemical energy released by the microbes' oxidation process is converted directly into electricity, rather than being lost as heat. That is a very high internal efficiency.
This raises an important question then. Why aren't these microbial fuel cells everywhere, cleaning all our wastewater and powering our smart homes? What are the operational challenges?
There are several critical interconnected barriers. The primary issues are power output and cost. MFCs suffer from notoriously low power density and low current output. You might have 80% internal efficiency, but the absolute amount of power generated per cubic meter of reactor is often too low to compete with conventional energy sources.
And what about the hardware cost?
High material costs are an existential threat. Effective electron transfer requires highly conductive electrode materials that must also promote robust biofilm formation. Historically, this meant expensive platinum-based electrodes.
And since the microbial community is complex, you also have major stability issues, right?
Absolutely. The sheer complexity of the microbial communities, which naturally evolve over time, leads to performance variability. You also have challenges related to pH fluctuations, changes in nutrient flow, and just the difficulty of optimizing reactor configuration at an industrial scale.
Let's turn back to the high-efficiency fuel production systems, the 17.8% biomass and 3.9% isopropanol systems. If the lab efficiency is so high, what are the biggest techno-economic bottlenecks for moving these fuels to the market?
The critical factor determining economic competitiveness is not just the maximum efficiency, but the volumetric productivity. This is the metric of how much product in grams you get per liter of reactor volume per hour. It's the throughput of the factory.
It's production speed and density over time, how fast you can fill a tanker truck.
Yes. And our sources show that boosting productivity is paramount. For these systems, simply increasing the baseline productivity from a low rate like 0.13 grams per liter per hour to a higher rate like 0.28 can reduce the overall biomass production cost by dramatic 23% to 38%.
And the other major bottleneck is the capital cost, or CapEx, especially for the EMP systems that rely on external electricity.
High capital intensity is the Achilles heel. Producing high-value fuels requires a huge initial investment in upstream components. Specifically, that means installing massive PV farms to power the electrolysis units or building complex photoelectrochemical systems that supply the continuous pressurized H2 to the microbes.
And cost modeling shows the difficulty of hitting price parity with current fossil fuels.
Correct. Modeling the production of fuels like N-butanol demonstrates that achieving a minimum fuel selling price, or MFSP, below $6 a gallon requires highly optimistic assumptions regarding efficiency and long-term performance.
An MFSP below $6 a gallon sounds ambitious, but that's still double or triple the current market rate for refined petroleum. How do the sources justify that price point, given the intense CapEx? Is the inherent storage capacity enough to bridge that economic gap right now?
That's where the analysis gets tough. Right now, the inherent storage capacity is the primary justification. These fuels offer immediate drop-in compatibility with existing jet engines and gasoline infrastructure, and they offer high density that batteries simply cannot touch for certain applications.
So the value is less in competing with cheap utility electricity and more in competing with premium, high-density petroleum products.
Right. Future cost reduction relies on achieving far cheaper PV units and further biological engineering breakthroughs that cut production time dramatically.
So we need cheaper catalysts that are also self-repairing and better engineered cells that last longer and process faster.
Precisely. We need enhanced system stability. Biocatalysts are inherently sensitive, and the inorganic materials can suffer from corrosion. Continued success depends on developing enzymes engineered for high tolerance and using robust, earth-abundant inorganic components, like the copy we discussed, which can even offer a self-healing capability.
And for photobioreactors, where we use cyanobacteria directly, we still have to fight physics, right? Like light management inside the dense reactor culture.
Absolutely. Reactor design has to contend with light attenuation. As sunlight passes through the dense microbial culture, it's absorbed, creating spatially uneven illumination. Cells near the surface get saturated, while cells deeper down are starved of light.
It sounds like we have proven the science beyond a doubt. 17.8% is phenomenal, but now we are fundamentally grappling with the industrial engineering of biology and electrochemistry.
We have moved decisively past proving technological feasibility. The challenge is now fundamentally economic. Bridging the gap between high bioelectrochemical efficiency in the lab and achieving sufficient volumetric productivity and system stability at an industrial scale that can compete with existing infrastructure.
So what does this all mean for you, the listener? We've covered three major pathways today that utilize synthetic biology to transform sunlight and CO2 into energy products, which we can collectively call the three H's of biosolar energy.
We can recall them as
First, high-octane fuels. This is the metabolic engineering breakthrough, taking cues from obscure Streptomyces bacteria to produce supercharged high-energy density molecules like fuelamycins. These promise energy densities of over 50 megajoules per liter, far superior to kerosene-based rocket fuel, and could revolutionize the aerospace industry.
Second, hybrid efficiency. This is the artificial leaf concept, which showed us that by coupling non-living, earth-abundant catalysts like that copy with engineered bacteria like Ralstonia eutropha, we can hit a record-breaking bioelectrochemical efficiency up to 17.8% for biomass production, a massive leap over natural photosynthesis and a viable route to stored liquid fuels.
And third, hydrogen hurdles. This is the photobiological approach using cyanobacteria to produce pure hydrogen, where the biggest hurdle is overcoming the O2 sensitivity of the hydrogenase enzyme. This forces species like cyanothes to use temporal separation daylight CO2 fixation and dark cycle H2 production, which synthetic biology is actively trying to override through enzyme engineering.
The future success of this field really hinges on continued biological engineering, increasing H2 tolerance, optimizing those promoters we discussed for precise resource allocation, and increasing that critical volumetric productivity metric. And that's alongside engineering design improvements to lower capital costs, utilize cheaper self-healing materials, and improve reactor stability.
I think we've shown that the age of the microbe as a precision fuel factory is absolutely upon us, moving beyond mere biomass to engineered performance
It
chemicals.
is. And finally, here's a final provocative thought for you to explore. The sophisticated metabolic engineering tools used today to create high-performance fuels like the fuelamycins are focused entirely on maximizing energy output. Have you ever wondered why we accept the low 1 to 2% efficiency of photosynthesis when it comes to food production?
Mm.
Could the advanced synthetic biology tools used here, like engineering artificial carboxysomes to increase CO2 concentration around rubisco, eventually be applied directly to agricultural crops? Engineering staples like wheat or rice to fix carbon with wildly enhanced efficiency, perhaps solving the food energy paradox using the exact same synthetic biology toolkits we are using to make rocket fuel.
A fascinating possibility to chew on. That's a deep dive for another day.
Thank you for joining again.